research. I'm going to look at genetic engineering, the diagram that you need to learn. How they'll ask you questions on this normally is they will jumble up the different stages. And what you've got to do is you've got to, in multiple choice, put them into the order, like A, B, C, D, and they'll give you definitions of what's happening at each stage, and you've got to put them in the order they come in the process. Also, in the paper 2, short answer section, you might get like a 3-4 word short essay on it, or diagrams where you might need to predict what's going to happen next, and not picking numbers like multiple choice, but being descriptive of what's happening at that stage. So it's not a very long chapter, okay? And the basics is, well, why do we undertake, or why is the need for genetic engineering? And the answer to that would be because it can mass produce desirable products in a very short space of time, okay? So we don't use organisms like multicellular organisms because, I mean, let's be honest, if you were waiting on a human to produce a desirable product, then it would take forever. So what we use is bacterial cells because they're so simple, and with that they are easy to divide, and in a very, very short space of time too. So that is the sort of point between it, and we can mass produce products such as insulin, which is probably the best example, like the most common example you'll get from genetic engineering, and how we can supply the prescription world with vast amounts of insulin using this process. So what we need to identify straight away is the actual gene itself, okay, which can transcribe and ultimately produce this desirable product. And we can get these genes from animal cells or plant cells, okay? So if you can imagine this here, okay, it's just a rough version of a cell. So if you can imagine the red being the nucleus, sorry, and being a rough, okay, rest of the cell, okay? So nucleus, rest of the cell. I have my elbows doing on it, that's why this is not taking too much. And within the cell, we've got, we've got chromosomes made up of genes, and I've highlighted the gene and the red desirable gene, which we are going to look at, and we are going to multiply to create this desirable product. So what we need to do at this stage, we'll keep going with the blue, right? We identify the desirable gene, and what we need to do next is we need to be able to separate that identified gene from the genetic information, right? So we're going to effectively cut it out, and with National 5, all you need to know at National 5 level is how to cut that out. You're going to use a specific enzyme which is going to allow us to cut out that gene from the chromosome, okay? So going on to next, yeah. So the enzyme action takes place there and there, allowing us to separate it from the gene. So effectively we've now extracted the desirable gene, and it's now available for use. And what then happens is we need to find somewhere to put it. So harken back to what I've just said, we're going to use a very, very simple bacterial cell, right? Bacterial cell, just to quickly refresh you, we have got a plasmid, we've got the chromosomal ring, okay, and this is just the bacterial cell itself, right? So the plasmid itself is what we're going to use in this, okay? So we're going to take that plasmid, but what we must do is we must get this desirable gene and place it into this plasmid. So we've cut this out using enzymes, and next we're going to extract the plasmid, so it's then going to be available for use. So we used enzymes, okay, to cut out the desirable gene from the original source chromosome, sorry. And what we're going to do next is extract the plasmid, and we're going to cut open the plasmid to allow this gene to be inserted in. So key thing to note here is when it comes to in National 5 the cutting, okay, so if you're cutting open the chromosomes to free the gene, or if we're cutting open the plasmid to create space to insert the desirable gene from the original source cell, you're going to use enzymes as well. So cut here, and you're going to have one cut there, is going to allow you to manipulate the plasmid or cut out that before. So enzymes being the scissors, keeping it simple with National 5, we're wanting to get that placed in there. Enzymes being the scissors, moves in to the plasmid. Enzymes are also going to be the glue, right? So key things so far: extract the gene using enzymes, cut it out using enzymes, extract the plasmid, cut open the plasmid to make room for the desirable gene using enzymes, and then we're going to paste the desirable gene from the original source into the plasmid using enzymes. So very, very straightforward at this point in time. So now we've got this new, I suppose, recombinant plasmid, okay, because you've altered the genetic information. The next thing we're going to do is go back to the very, very start, and you're going to put the plasmid back into this bacterial cell. So you've manipulated it, you've put in the new source gene, and placed it back into the original, bacterial cell. Why bacterial cell, as we said? Because bacterial cells are very, very good at duplicating in a very short space of time, and then that allows us to mass produce this desirable gene, and ergo the product that comes with it. So once the plasmid is placed back into the original bacterial cell, it's just then grown under ideal conditions, and it's just going to be allowed to divide and divide and divide. So before you know it, I mean, the lifespan or the time taken for a bacterial cell to divide and create identical copies of itself, two identical copies of itself, is very, very short. So of the new bacterial cell, okay, I suppose it's the old one, but it's got the new information in it, you go quickly from one bacterial cell, and then we go to two, right? So we've doubled that, and then the two identical cells are then promoted to divide again, and then you have got 4. And you don't stop at 4, you keep going, you keep mass producing, you keep dividing the cell into 2 new identical cells. So we go from very, very quickly, we went from 1 to 2 to 4. 4 is going to go to 8, 8 into 16, 16 is 32, 32 to 64, 1, 2, 8, 2, 5, 6, and so forth and so on. So before you know it, you have got access to cells that can create this product through this whole process of genetic engineering. Another thing you have to bear in mind is that genetic engineering is usually for the benefit of man, okay? So as well as rapid times to create duplicates of the identical bacterial cells, it also saves a lot of time and money, okay? So genetic engineering, using a bacterial cell to mass produce a certain product using a gene extracted from original source, and it's going to save you time and money and ultimately a lot of effort once you've done the original cutting and pasting with the enzymes. Okay, so National 5, very, very short and sweet today. And if you find this useful at all, then if you give it a like, a thumbs up or whatever, leave a comment. Also, if there's any requests you want me to do, I can do that moving forward for you. But in a nutshell, that this morning is genetic engineering at National 5. Tomorrow morning, we're going to look at nerve impulse transmission at advanced higher level 1.4D section I. Okay, I'll see you then if you're doing advanced higher biology.